Good morning, all you Soo High AP Biology students. This is Mr. McLeod again. Welcome to our next podcast on analyzing the evolution of populations using the Hardy-Weinberg equation and method. Today we'll briefly review Hardy-Weinberg equilibrium and then learn how to analyze a population and use the data to determine whether or not the population is changing over time or not, otherwise known as evolving. So get your notes out, some to write with, and a calculator, and let's analyze. Now you may recall from your summer assignment that there are only 5 real major agents of evolutionary change that are commonly recognized by biologists. Let's get those back in your notes for review purposes. As you can see in this slide, those agents are mutations, gene flow, non-random mating, genetic drift, and natural selection. So get those in your notes right now. Now it's important that you understand that these are the 5 agents of evolutionary change, because we're going to be talking about them in some detail here pretty soon. Also recall now what a population is. Make sure you recognize that for a population to be actually considered a population, they must be interbreeding. Remember, it's a group of the same organisms that are interbreeding in a particular area. Now this eliminates any kind of hybrid that can't produce viable offspring like mules or walleyes, for instance. So get that in your notes. Also recall that a gene pool is a collection of all the genes in a population, both dominant and recessive alleles. Also write that down. It's extremely important that you know the difference between a gene and an allele. Remember that a gene consists of 2 alleles. They can be homozygous dominant, where both are dominant alleles, homozygous recessive, where both are recessive alleles, or they can be heterozygous, aka hybrids, where one is one dominant and the other a recessive. Yep, get those in your notes for review. And then lastly, recall that allele frequency is how common that allele is, dominant or recessive, in a population. Also get that down. Now, if you see in this slide, an example might be plant height. How many capital T's or dominant tall alleles versus how many lowercase t or short recessive alleles in a population are what we're talking about for allele frequency. So make sure you get that in your notes and you understand those 3 concepts. Now, by using allele frequency, we can now look at evolution as the change in the frequency of alleles in a population. So let's ask ourselves a key question here. What conditions could cause the allele frequencies to not change? In other words, what would the characteristics of a non-evolving population be? In effect, we'd be removing all 5 major agents of evolution, right? In other words, we would have the following in your notes. 1, a very large population size, which means no genetic drift. Make sure you're getting these down. There would be no migration, and that would prevent any gene flow in or out of that population. There would be no mutations, which would prevent any genetic change over time. There would also be no random mating, which prevents any sexual selection. And finally, there'd be no natural selection, which means all organisms would be equally fit in their environment. So make sure you get those down. It's important that you understand what the characteristics would be of a non-evolving population. Think of the non-evolving population as a control group. And remember the purpose of the control group, which is to compare results. So think of it that way. Okay, now on to the Hardy-Weinberg principle. G.H. Hardy, the English mathematician, and W. Weinberg, the German physician, independently worked out the mathematical basis of population genetics in 1908. Believe it or not, I'm going to have you write that down because that may appear on the exam. Now, their formula predicts the expected genotype frequencies using the allele frequencies in a diploid Mendelian population. Get that in your notes. So their formula predicts the expected genotype frequencies. That's the frequencies of a particular allele in a gene pool in a diploid Mendelian population. That's a typical two allele population. Now, they were concerned with questions like, what happens to the frequencies of alleles in a population over time? And would you expect to see alleles disappear or become more frequent over time? Now, their concept of equilibrium applies to a hypothetical, non-evolving population, which serves to preserve allele frequencies. Write that down. A hypothetical, remember what the word hypothetical is, kind of an imaginary, non-evolving population. That is not a typical natural population. So their idea was to use the hypothetical, non-evolving population as a model that they call the Null Hypothesis. Write that down. This non-evolving population is in what we call Hardy-Weinberg equilibrium. Hardy-Weinberg equilibrium. You see it right here. A hypothetical, non-evolving population that serves as a model we call the Null Hypothesis. So make sure you get that in your notes. It's extremely important for you to understand that Hardy-Weinberg equilibrium is a hypothetical, non-evolving population. It's not real, it's not typical in the real world, but we're going to use it for comparison purposes, much like a control group. As you might have already figured out, models rarely mimic what really happens. This model's no exception. Natural populations are rarely in Hardy-Weinberg equilibrium. As we said, the Hardy-Weinberg model we're going to use to measure if forces are acting on a population. Get that in your notes. We're going to try and figure out if something's going on to change the frequencies. So think of HW equilibrium as the control group in which we can compare all other populations against. We are actually measuring evolutionary change by comparing a population model that is not and cannot change. Also write that down. We call this non-evolving population our Null Hypothesis. Make sure you understand that the Null Hypothesis is what we'll compare now the population changes with. So make sure you get that in your notes. Okay, now let's get into the nuts and bolts of this concept. The Hardy-Weinberg theorem, which you see here, involves counting alleles. In the example in this slide, we are assuming two alleles. Big B for the dominant allele for black coat. It's not really black, but it's dark. You kind of get that. And little b for the recessive white coat color that you see here on the bottom right. Now, make sure you get that down. So big B for black, little b for recessive white. Now, in the Hardy-Weinberg theorem, we'll let P, little p, equal the frequency of dominant alleles, or big Bs. And we'll use Q for the frequency of recessive alleles, or little b's. Also write that down. Now, it should make sense to you that P plus Q should add up to 100%, or all of the alleles possible. So we'll let P plus Q equal 1. We'll say 1 is 100%. So make sure you get that down. P plus Q equals 100%, and we're going to let 1 equal 100%. So P plus Q will equal 1. Make sure you write that down. Now let's apply these concepts to the actual genes. Remember, genes have pairs of alleles, or two alleles. Homozygous dominant, homozygous recessive, heterozygous. So first would be homozygous recessive, or homozygous dominant, or two dominant genes. Now that would be represented by P times P. Right here, P times P. Well, if we multiply P times P, we get P squared. So write that down. So P squared is the frequency of homozygous dominant genes. Well, if P squared is the frequency of homozygous dominant, then Q squared is the frequency of homozygous recessive genes. So write that down. Now what's left is the frequency of heterozygotes, which would be P times Q plus P times Q. What we get from that is 2PQ. So 2PQ is the frequency of heterozygotes. Write that down. Now also remember that all these added up, P squared, Q squared, and 2PQ, must be equal to 100%, or 1. So here's our Hardy-Weinberg equation. P squared plus 2PQ plus Q squared equals 1. That's the one you're going to use to answer these problem questions and on the exam. So get that in your notes. So when we're solving Hardy-Weinberg problems, it's important to understand two things. One, P plus Q equals 1, and you'll see why that's important here pretty soon. And then 2 is P squared plus 2PQ plus Q squared equals 1. So make sure you have those two equations handy when we start doing these problems. Okay, now let's use an example. Let's say that we have a population of 100 cats. 84 are black and 16 are white. Remember, we're going to set up our Hardy-Weinberg equilibrium, or the one that's not evolving. And then we're going to compare now the next generation to this one to see if evolution is happening. Now, what we can't tell is which of the black cats are homozygous dominant or heterozygous by looking at them. They both look black. So the question is, how many of each phenotype will there be? So that's the question we're going to ask. How many of each genotype will there be? Remember the difference between phenotype and genotype. Genotype is the gene combinations, the allele combinations. Phenotype is what the cat looks like, black versus white. We want to know how many of each genotype there are. So make sure you understand that. Now, as always, we need to work from information we know first. Now, of all the information given, we can absolutely know that 16 of these cats are homozygous recessive, or white. It says it right here. There's 16 whites. We know for sure that their genotype is little b, right? So we know that 16 of those are little b, little b. What this means then is that 16 out of 100 of these cats have the phenotype little b, little b. Remember, phenotypes, what they look like, genotype is their allele pairing. In terms of our equation, this means that q squared equals 0.16. 16 divided by 100 is 0.16. So q squared would be 0.16, which you see right here in this slide. Well, if we know that q squared is 0.16, we can then figure out what q is, correct? If we know q squared is 0.16, then the square root of 0.16 would give us q, and that would be 0.4. Make sure you get that in your notes as well. Okay, now that we have q, you should see that it's pretty easy to figure out what p is. Remember that p minus q equals 1. Well, if q is 0.4, what do you think p would be? So p then would be 1 minus 0.4, or 0.6. Make sure you get that in your notes, and make sure you understand how we got that. Okay, so we now know that p is 0.6 and q is 0.4. Once we have p and q, we can then go back and plug these values back into the Hardy-Weinberg theorem equation. So in this case, p squared would be 0.36, right? 0.6 times 0.6 is 0.36. 2pq would now be 0.48. So that's p times q times 2, which would equal 0.48. And q squared now would be 0.16. Right? 0.4 squared would be 0.16. Get those in your notes and make sure you understand how we got all three of those numbers. Now you can always check your work by adding these values up. 0.36, 0.48, and 0.16 better equal 1, right? P squared plus 2pq plus q squared always needs to equal 1, and in this case they do. Now we can assume that this, or we'll use this population as the Hardy-Weinberg equilibrium. We're going to assume that this population now, all right, is not evolving. What we're going to do then is look at the generation of cats that this population produces and look at their frequencies. Once we get their frequencies, we can now compare them to these frequencies. If they're the same, what do you think that means? It means that their population is not evolving. If somehow they were different, it would mean something's going on to change the frequencies, therefore that population is changing. Make sure you understand that in your mind before we move on. Now, in our original sample now, 84%, 30.36 plus 0.48 are black, and 16%, 0.16 are white. Does that all make sense? So now, again, what we're going to do now is compare the next generation results. So let's do that. Now, here's our Hardy-Weinberg equilibrium generation right here. We now have those cats right here. 36% p squared, 48% 2pq, and 16% q squared. Or we could say 36% are homozygous dominant, 48% homozygous recessive, 16% homozygous, or 48% heterozygous, and 16% homozygous recessive. Now, remember, because they're in Hardy-Weinberg equilibrium, we call this our null hypothesis. What we want to do now is look at the second generation and see what's happening with them. Okay, now here's our second generation cats, we call sample data. This data now we're going to compare with our Hardy-Weinberg equilibrium to see if anything's happening with this population. In our original sample, the hybrids appear to be weaker in the second generation. The hybrids, that's these, appear to be weaker in the second generation. Do you understand? You see that? So 48% of them are hybrids in our Hardy-Weinberg equilibrium, and now only 10% are hybrids. So they appear to be weaker, or something's going on with those hybrids. And our job is to explain how you explain this data. So stay with me here. Now, since we can clearly see that something's going on to change the frequencies, we need to ask ourselves what's causing the population to appear to change or evolve. Now, we're going to hypothesize what might be causing it. Now, where do we get that hypothesis? From those 5 factors you listed below. Something, some one thing or combination of those 5 things is changing in order to make these frequencies change. So our job is to hypothesize what might happen to change the frequencies. So you got to ask yourself, which of the 5 characteristics of a population in Hardy-Weinberg equilibrium could be responsible for this? Any ideas? Think about it. What's most likely probable is an immigration from an external population that is predominantly homozygous big B, big B. Now, how do I make that assumption? Well, look what happens to the homozygous dominants. They go from 0.36 to 0.74. Well, how does that happen? Well, one of the ways that could happen is you get an immigration now, in this case, of black cats that are big B, big B. Does that make sense to you? I hope you're shaking your head. Yeah, I guess that would make some sense. Now, can you think of anything else in those 5 that might happen? Now, it could be that there's some non-random mating occurring. Now, here's what I mean by non-random mating. That all of a sudden, for whatever reason, black cats only breed with black cats, and white cats only breed with white cats. Now remember, there's far more black cats than there are whites. So if black cats start to breed with blacks and whites with whites, then we're going to end up with more black cats and therefore more big Bs, bigger big B, it sounds like the coffee place, frequency than we did in here. So if these cats decide to only breed with blacks and these only with whites, then we're also going to get a corresponding increase in the frequency of dominant Bs. Does that make sense to you? Hopefully you're shaking your head. Yeah, that makes sense. Okay, let's bottom line here. The bottom line here is that all we can say about the second generation population is that it's changing and not in Hardy-Weinberg equilibrium. In Hardy-Weinberg language, we would not accept the null hypothesis. In other words, we would not say that this population as a whole is in Hardy-Weinberg equilibrium because the frequencies are changing so dramatically. So the second population is most definitely changing or evolving for whatever reason. Does that make sense? Hopefully it does. Okay, now that's it for the Hardy-Weinberg introduction. Submit your notes to Moodle, then go get the problem worksheet on Moodle and start doing the sample problems. We're going to go over them in class this week. You can use your notes for this podcast to help you. Bye.